So we are talking about the Agrobacterium mediated gene transfer. Agrobacterium tumefaciens is firstly it is a gram-negative bacteria with rod shape, and it is found in soil, and it infects plant. Agrobacterium tumefaciens, it infects plant, especially the diocote plant. It does not infect monocote. This bacteria transfers its genetic material to the plant, and suppose this is a plant, then this root region, the area surrounding the root region is rhizosphere, and the area surrounding the shoot region is phyllosphere. So this penetration process, it occurs in the rhizosphere region. So in the form of genetic material, what bacteria actually transfers is Ti plasmid. Ti plasmid is tumor-inducing plasmid. So this tumor-inducing plasmid, it will be transferred to the plant, and this tumor-inducing plasmid is like around 200 kb. And like what actually it is like bacteria has its chromosome. Then apart from that chromosome, it has extra chromosomal circular thing that is called as plasmid, which is self-replicating and is extra chromosomal. So these plasmid acts as a Ti plasmid, and which causes crown gall disease in the plant. So there is another bacteria called Agrobacterium rhizosine, and it transfers Ri plasmid to the plant and causes hairy root disease in plant. So R stands for root. So the Agrobacterium tumefaciens, it causes crown gall disease, while Agrobacterium rhizosine, it causes hairy root disease in plant. And this Agrobacterium rhizosine is also a gram-negative bacteria. Suppose this is a Ti plasmid, then it has regions called origin of replication, ORI region. Then it also has VIR regions, virulence region, which contains virulence gene, and it is necessary for the transfer activities. Without this virulence region and virulence gene, the transfer is not possible. Then it has tDNA region. On the tDNA region, it is bordered by left border and right border, and these left and right border are of 25 base pair each. And this tDNA contains phytohormones like genes for secreting phytohormones like auxin, cytokinin, and opine. And after moving into the plants, overproduction of these phytohormones, auxins and cytokinin occurs, which will lead to the formation of tumor. And if the right border is absent, the transfer process doesn't occur. Right border and virulence gene, also the origin of replication, all these are necessary for the process of transfer. So some of the opine catabolism gene includes ONFMA. So O for octapine, then nopalin, then fructopine, manopine, and agropine. So these are some of the catabolism gene, opine catabolism gene. Similarly, TRNR for cytokine and TRNS1 and 2 or OXA or B for auxin synthesis. So for the process of transfer, three things helps. First one is the border region, second is virulence genes or virulence gene region, and third one is chromosomal virulence gene. The third one is present in the third one is located at the chromosome of the bacteria, that is chromosomal virulence gene. The virulence gene, these are present in the form of operon. No, so operon, it basically consists of promoter, operator, and structural gene. Operon does not consist of regulatory gene. So promoter, operator, and structural gene. And this virulence gene consists of operons such as Vir A, B. B has its many subunit, C, D, D1, D2, D4, E1, E2, F. So the damaged plant, it releases phenolic components in the soil, mainly acetylcysteine, which then travels to the bacteria, and the Vir genes of the bacteria, it senses those phenolic component and then activates the further processes. So these operons Vir A and Vir G, these are constitutively expressed in bacteria, which means they are expressed all the time. And once the acetylcysteine like enters the bacteria, Vir A gene recognizes it, and in the process, the chromosomal virulence molecule CHVE, it helps Vir A to do so. And after that, what happens is that this Vir A, it autophosphorylates itself. So it adds it acts as a sensor kinase or histidine kinase or sensor kinase, as it sends acetylcysteine. So it is called as sensor kinase, and kinase because it adds phosphate group. So it does autophosphorylation on its histidine amino acid. It has histidine amino acids on it, so it phosphorylates itself at histidine amino acid, and then what it does is like it transfers that phosphate group to the Virg operon, and in Virg there is another amino acid aspartate, so it phosphorylates Virg at aspartate. And then this Virg is called as a response regulator because it is responding to the like Vir A. And then again and also it further activates other virulence genes. Like it activates vir D, E, F. So further it activates other virulence gene. So now there is this operon VIR-D2. VIR-D2 is a endonuclease. So endonuclease, it cuts the DNA in between. So it cuts the third or fourth base pairs uh, of tDNA at right border of the lower strand. And then this there is this VIR-D1, which is a topoisomerase. So it helps VIR-D2 to recognize and clip within the border sequence. And there is this enhancer element present near the right border. Now with C2 and C12, they come to that place and they recognize this enhancer element and finally enhances the cutting process which is being carried out by with D2. So once the cut has been done, now the tDNA starts separating and the new strand starts synthesizing on that cut. So once the synthesis has been done, the tDNA it separates out, and now we have the separate tDNA. Now we have the separate tDNA, and at the suppose this is 3 prime of the tDNA and this is 5 prime of the tDNA, still at the 5 prime side of the tDNA, like this weird D2 is still attached to it. So how it is still attached to this 5 prime end is because of the tyrosine amino acid present in this weird D2. It has this tyrosine amino acid, so because of this it is still attached to the tDNA. Now, suppose this is your bacterial cell and this is your plant cell. So the tDNA has to transfer from this bacterial cell to the plant cell. So here, in between the two, a channel is being created. Transfer apparatus is being created. So suppose this is that channel, and this channel is called as type 4 secretory system or type 4 secretion system. And this channel is formed by VB, and VB has its 11 subunits, and Vir D4. So Vir B and Vir D4. This Vir operons, they intercalates with lipid to form this channel, which is called as type 4 secretion system. Once this channel has been formed, and we know that in this bacterial cell, the tDNA has already been separated, and with its in its five and the tDNA in its 5 prime end there is weird D2 bound to it. So now that tDNA is transferred via this channel to the plant cell, and along with tDNA, this weird D5, E2, E3, and weird F, these are also transported or transferred individually into the plant cell. So along with tDNA, weird D5, E2, E3, and F, these are also transported or transferred to the plant cell. And once the tDNA is inside the plant cell with D2 at its 5 prime end, now it has to transfer into the plant nucleus. So here the VE2, it shows DNA binding activity, and now the tDNA along with VE2, this all tDNA with D2 and E2, these are called as T complex. Now this has to be transferred again into the plant nucleus. This vir E2 and D2 has nuclear localization signal, so they can carry the whole complex inside the nucleus. And inside the plant nucleus, plant has its genome. Now the T complex is also there. So now the T complex and plant genome has to be integrated. The T complex has to be integrated into the plant genome. So for that, non-homologous recombination occurs between the T complex and plant genome, and it is integrated into the plant genome and finally causes tumor formation. So before integration of T complex or tDNA into the plant genome, the tDNA is single-stranded. No, so first it gets converted into double-stranded tDNA, then only it integrates into the plant genome. So as a whole, while talking about Vir-A, it acts as a protein kinase or sensor kinase or histidine kinase and recognizes or senses the phenolic component acetylserine, which has been released by the damaged plant. And then comes Vir B1, which has its subunits, still Vir B1 to B11 and Vir D4. Vir B1 to B11 and Vir D4, they assemble and forms the channel or the transfer apparatus, which is called as the type 4 secretion system. And then Vir D1 is there, which act as an endonuclease, and this Vir D1, it act as a No, I'm sorry. With D2, it acts as a endonuclease, and with D1, it acts as a topoisomerase and helps D1 to recognize and cleave within the border sequences. Then with D2 is a endonuclease, so it cuts the third or fourth base pairs of tDNA at right border at lower strand, and then after that, there is this Vir C2 and C12, which enhances the activity of the Vir D2, the endonuclease activity. It enhances then by binding to the enhancer present near the right border. And then this Vir E2, it has nuclear localization sequence, and Vir E1 is also there, which Vir E2, which also acts as a SSB, a single-stranded binding. Then SSB, but is non-specific. So and then it also uh, protects it from the nuclease attack, and this Vir E1, it acts as a chaperone and stabilizes Vir E2 in the plants. And then comes Vir H, it provides host specificity, and then Vir F is there. So Vir F, it promotes proteasomal degradation of BIP1 and BIP2, which are basically the plant bacterial defense proteins. So I was a bit confused in that, like, transfer transfer wala part. There what happens is that Vir D2, which is bound to the 5' end of the tDNA, so that Vir D2 bound tDNA, and Vir E1 and E2 and Vir F, they move to the plant cell via T4 secretion system. Then once it enters the plant, Vir E2, which acts as a SSB, it binds immediately to the single stranded tDNA to prevent its degradation from the nucleases. Then Vir E1 acts as a chaperone, so it helps E2 to do so. And then there in plant, what happens is that the flagellin binds to the plant rich repeat leucine rich repeat LRR receptor. So flagellin binds to a plant leucine rich repeat LRR receptor, which triggers the plant MKK4 or 5 signaling pathway. After that, the after the completion of the pathway, the end product is phosphorylated BIP1. In normal case, that phosphorylated BIP1 enters the plant nucleus and stimulates the expression of pathogenesis related bacterial resistance genes. Okay, uh, pathogenesis related bacterial resistance genes. But the tDNA with D2 at its 5' end along with E2 at somewhere binds to these phosphorylated BIP1, uh, and along with that BIP1, they all enter the plant nucleus. Now this T complex is inside the nucleus. Now the VRF, it degrades phosphorylated BIP1, and also um, and thus preventing expression of the bacterial resistance genes. So once BIP1 is degraded, there will be no production of the bacterial resistance genes. So once the work has been done, VRF also remove the bound BIP1 and VE2 protein from the tDNA by proteasomal degradation, and then tDNA get inserted into the plant genome, and then synthesis of auxin, cytokine, and opine occurs, and then the tumor formation occurs. So the overall thing is that this GI plasmid is used to introduce new genes into the plant cell. So for that, the gene of interest or the new genes has to be inserted into the tDNA, and bacteria then can integrate those into the plant chromosomal DNA, and it can be expressed. So for the process, the binary vector strategy and the co-integration strategy can be used. In binary vector strategy, two vectors are used, vector 1 and vector 2. Vector 1 consists of plasmid with Vir genes, and vector 2 consists of plasmid with tDNA, but not Vir genes. So this is binary vector strategy. And the co-integration strategy, it uses an entirely new plasmid. Then there they use the E. coli vector, but that E. coli vector carrying a small portion of tDNA, so it only uses one vector.